I'm reminded that I hope the alcohol I drank last night kills any sepsis I have. I also reminded myself that I have to call my wife, who just texted me about her second UTI in the last month. I'm definitely going to buy a new toothbrush. And I will tell you that I am old, if you don't know. Being a senior consultant in my world just means that I work less and get paid more money. But senior means I'm old. And I reflect back real quick from what John said, having worked in things with, SARS-1 in Hong Kong and in Canada, things that I've done in my life with COVID, that this has been impactful. And I just wanted to bring it up because, frankly, I'm not going to talk about any of that at all. So anything that I think I did that was important, I'm going to talk about this now. And that is important to me now because it has a premise of infection control back to device manufacturers. So all the things you heard this morning, this is a completely different topic. So I like to think that I'm a storyteller, and I like to also say that if I'm finishing things up, I'm also the comic relief for this session. I know very well that I'm preventing anybody from getting a shot of coffee, and the vendors hate me because if I keep you too long, you don't get to visit their booth. So we'll go into this. I'll definitely get the time that I get going. I saw John's trick when he put his timer on, so I put my timer on as well. But I'm going to talk about something that I have learned. This is a story, and if I use I, it's not an I meaning that it's something that I'm talking about specifically about me personally and wanting to what I learned, but it's a story. I'm going to tell you a story, and any of you that have had an issue, maybe I should have a raise of hands. Has anybody ever had an issue in the OR with a dirty piece of equipment that wasn't cleaned properly? Yeah. So this is not a topic that anybody needs to talk about, right? I've built devices for almost 30 years, and I've designed them. For the past 15 years, I've actually designed cleaning chemistries. So I've seen it from both sides, and I've been challenged with trying to figure out, why can't I solve the problem? Because people say, you're building these things, you're supposed to clean things, they pass all the regulations, and you're saying, why isn't it working? So this is where it comes down to expectations versus reality. I'll tell you, why do I have such a negative opinion of enzymes? So when I started on this, I started looking at all the cleaners I had, and why did I start? I'd like to say I start because I was a very inquisitive person and I wanted to solve a problem. I actually got started because the FDA wrote me up. As you can tell, I'm from the States. I apologize for my accent. I also am from the state of Michigan. I am retired, that's why I'm a consultant. Just to let you know, not everyone in the United States that retires lives in Florida. I actually live in Michigan. You can always tell someone who lives in Michigan because when you ask them where they're from, they'll do this. And you say, where are you from? I'm from here. Anybody who knows Michigan, Michigan's shaped like a mitten. And when you talk to someone from Michigan, they say, where are you from? I say, I'm from over here. Oh, you're from Detroit? Oh, yeah. Well, no, I'm from Mackinac. That's up here. So if anybody from the States and you ask them, you say, where are you from? And they show you your hand, where are they from? Michigan, okay. Just want to make sure that you all know where I'm from now. So when I work for a company that has many people that are from North Carolina, I do not talk like this. But the reason I got into this was I worked with the FDA. The FDA wrote me up because I used to work with power equipment. Anybody know what power equipment, sawbones and things like that? What used to happen was the FDA wrote me up and said that you have leaking things coming from your equipment that's getting into your patients. We need you to solve that problem. And they said it was oil. And I said, I don't have any oil in my devices, so what the heck it's not my problem. Someone else must be doing something to it. I said, that's good enough. You're a big company, you need to be able to fix it. So we started to look at why we had those issues. And that's why I started to study some of the things that I have to talk about. Well, I told you that I used power equipment, right? Well, this is a piece of power equipment. It's a chuck, or a chuck that holds a reamer. And I can tell you that everything you see in there that's black and little particles is a contamination. None of it's biologic. Zero. What you're seeing there is contamination that is judged by degradation of material, conversion of grease materials, or remnants left over from detergents. And that's why I studied them. I spend a lot of money analyzing all of these types of materials and what they are. So what I decided to do was I started to look at detergents. I took them apart, or listed all the things that they had in them, such things as surfactants, builders, pH balancers, anti-corrosives, everything. Listed them all down, the components, the enzymes, everything, took them out, and started to build something back that would clean and not have anything extra nor need anything extra in it. So as you can see, I was real happy because the past, if you look at that past, that is a it's a very big picture of a very small bearing. But in that bearing is some grease. That grease doesn't move. It's like the kind of grease you have on your car tires for brakes, or not for brakes, but for the car tires to move around. But when you affect this with different chemistries, the grease changes. And what you're seeing here is in the past picture is the liquid oil portion of the grease has been converted by the detergent chemistries into its base components, which is basically a clay base, an oil base, or a dye. So this is basically sand. So what happens when you put sand in a bearing? Not very good things, right? They break and they work. Well, I made a brand new formula. I took out the enzymes, those bad enzymes. I took out all the builders, I took all the pH balancers. I only used a couple of really good surfactants, and I solved my problem. There they are, I'm done. Well, all of a sudden I wasn't done, because I made this product and I sent it out to all of you folks to use. You come back and you said, it's still not cleaning. It's like, well, what's the problem? I mean, I'm testing all the things I'm supposed to do from a validated state, from surrogate challenges. Well, the problem when you use surrogate challenges in a laboratory is you may not actually mimic what's clinically available. So what we did was we started using soiled instruments. These are instruments that are single use. You know, so you can pull them out of the trash and you can test on them, but you don't have to, reuse them at all. And we weren't interested in reusing them. We were interested in doing study to say, what can we get real clinical soil off? Because honestly, real clinical soil was not reflective of the surrogate soil that we tried to clean for validation. So after we did this, we found out that any numbers of cleaners, 1 through 10, I'm not going to tell you which ones they are. I know what they are, and I'll take bribes if you want. Let me tell you what they are, but no, never mind. You can say that from protein removal, some did as much as 75%, some did as little as 20%. And all of these are sold as cleaning medical devices. Rather shocking to me from the fact that there was such a diversity. So I looked at mine, and I'll tell you mine's not on this list, but it's on the other side, so it does really well. But then I started to say, well, why do these other ones all have enzymes in them? Everybody wanted an enzymatic detergent in the US. Well, when I started looking at the enzymes, I'm like going, well, I don't need any enzymes. Because look at the factor here. There's the same 10 detergents. Some of them are up there, and they have very little use at all. You come from these types of things, you say after you're cleaning them, they look just as clean or as dirty as they should be. One thing I will say is, the ones on the bottom here, this might surprise you. If you believe me or not, those all pass the required numbers of protein residuals for cleanliness. Anybody think it's a little shocking? Do they look clean? So it was shocking to me as well. So I was feeling pretty good. I had my detergent, everything was fine. I was working well. I had one that worked well. I didn't have any enzymes in it, only had a couple of things, very simple. Everything was working great. And then a 180-degree turn. And I'll tell you, it's here because the company I'm actually been sponsored to present for this Novozymes, an enzyme making company. They don't make solutions, they make enzymes. Basically, they came and they said, well, our enzymes work, and I said, no, they don't. I got proof, I haven't used it at all, my stuff works great. And they said, well, that doesn't meet our data, because our data says that they should work better. If you look at the literature clearly defines the fact that if you use enzymes in a product, it cleans better. There's all kinds of other conjectures of what enzymes have as other consequences of their use, but those are very, very well studied by the enzyme makers. So they convinced me to try and do some more study. What I found was if you actually use the right product with the right concentration in the right dose, you can effectively clean things, very, very, very well. If you look at there, we're dropping. No, these are not cleaning tests where we say they should be at 0. The cleaning tests are things that you can do consistently to get the comparable numbers from different types of detergents. So with an optimized enzyme, I was able to pull almost all the protein off of these products. Now, this particular product is a single-use blade that gets thrown away, and it's got real clinical soil on it. And it's just the numbers we found on them, as much as 255 when it came out, and as little as 9 after just a simple soak and sonication with an enzyme. So I was completely convinced at the time that I could be able to add the right enzyme back to my detergent and it would work. And my story was like, okay, there are some unique issues with what I make. I make things that have grease in them. So I don't want it to affect the grease, but I want it to clean the clinical soil off. Well, then again, I test 5 more different detergents, and quite honestly, you can see one on the side that has a dye. It incorporated the dye, but it didn't it didn't hurt the grease. The one on the end didn't hurt anything, but the 3 in the middle completely wiped it out. So again, these are all the same detergents. Those detergents you get, they say, hey, this cleans medical devices, it's clear for metal, glass, whatever, but it has problems. Some of these things have to be able to be demonstrated to say why they can do these types of products. So the whole purpose was when we were trying to do this was imagine a case where everything that you have in your purview to use to clean the products actually has improves your efficiencies, improves your process, makes sure that you can get your existing processes done faster, and that you can use the right products and the right formulas in any kind of new aspect to make those 2 situations happen. Now, the second blades, I would say, are clean. Well, we all know that these are flat pieces of steel, and most products that are used for testing are done on flat coupons. But do we have flat coupons when we use things in the ORs and in, they look like this. Now, I put these up here not from the standpoint that they're reprocessable devices, but they're single-use devices that I can test that had clinical soil on them. So when they came out, they looked like the far side. They look like things you've seen before. Yes, and nod your head. They've also sat for about 4.5 months. That's something you don't usually see, right? We get them shipped to us, we keep them in a biohazard bag, and we put them in a biohazard lab, and then we study them from that perspective. But what ends up happening is when we use the right formulas and the right chemistries, and we use the best products that we can have available to us, even things that have the right enzymes that are clinically optimized, we can get these things fairly clean. And I can tell you that one of the challenges I had in a lot of cases was anything that's white, as you saw with those blades earlier where they said they passed everything, but they still had specks of dirt on them. When you make something that's white and it's plastic, a little speck of something doesn't pass anybody. Any of you guys would let anything in that has a speck of brown something on a white handle? No, it's got to be completely clean. The other interesting part we found about this whole thing was that after we had finished the whole process, we had a 45% reduction in time. So not only was it better for cleaning, it reduced the time necessary to do it. Now, am I saying that's a wonder drug? Here, use this one soap and does everything? No. What I'm trying to explain here on a test is that, or on a story, is that there are many, many, many things out there that are designed and are made and are promoted to help you do your job as far as cleanliness goes. Many of those things need to have more proof and more study and more test, and you should be asking for it in order to confirm that it's going to do the job that you need it to do. So where do I see the future going? How many of you are doing manual cleaning? Only. I can tell you that I can manually clean something better than you can do it in a washer. But somebody next to me probably can't. You know, it's a concept of manual cleaning is done well, can be done and actually clean sufficiently. The issue is it's validating its consistency is extremely hard. Doing a washer and having a washer be consistent, you can at least control the parameters of the washer. So what you want to be working for in the future is those same chemistries I'm asking you about to verify. Ask them, how they work in the washer setting and how can those be worked to work as efficiently as those simple tests I showed you earlier when they come out of a washer. So a lot of people use detergents. If you look at your detergent, and I will give you a sidebar that I just have a pet peeve about, is that if the detergent says something like, use a 1/4 ounce a gallon for manual and use an 1/8 ounce a gallon for automated. You can all see that nice little circle over there. It's like when you did your dishwasher at home, do you use the same detergent? You know, a little drop of liquid dish detergent in your dishwasher. I've done that. My wife doesn't like me anymore if I suds out the entire kitchen. But the chemistries are different. So when you use things in an automated system, the chemistry should be different. It's the way they're supposed to work. So ask those questions like, why does this one work in the washer better? Washers become the normal. They will continue to be the normal. What I struggled with when I was a manufacturer was you guys didn't want to do any manual cleaning or any manual pre-cleaning or any manual brushing or any manual rinsing or any manual anything. You wanted to take it straight out of the OR and put it in the washer. You remember that first picture I showed of an orthopedic chuck? You can't do that. The washer doesn't move the chuck. So you at least have to put it somewhere in some kind of water or solution and move the chuck so you can get something on that mated surface. Or think of it in the future of having washers that move it for you. So that's where I would see the future going. It's not happening today, but if you want to clean those types of mated surfaces, you have to do some type of manual process, at least to get them wet. So if you want to treat everything, you want to make sure that everything's designed to do all of your general cleaning. There is really no one solution for everything, so be careful with that kind of concept. But what do we need to do better cleaning? And I will finish up with this to hopefully keep us all on time and everything's going great. They're not going to prevent you from getting coffee, but education is one of the biggest things. That's what I'm here for today. Any of you that were harassed by us that were in our booth, you knew that we were like just standing in front of you pushing little files that said, do this education. That's all we're trying to show from the Novozymes perspective is we're trying to educate people that there are enzymes out there. Safety profiles, people talk about, allergies and things like that. I mean, there are many, consumer-based enzymatic products that have very, very low admittable standards for aerosolization, as low as like 15 nanograms. So it's time to take another look at all the processes that you have available to you and see if you can incorporate them in. Check the device compatibility. I'm always reminded of the young lady that I trained in my old job who then now did all of the work on the Da Vinci robot. She was very adamant about making sure that the cleaning chemistries would actually work and be compatible without appreciating the function of the device. As you know, with my products, it's like if we remove the grease, the device isn't going to function. Invest your best practices. And the best one I would say is ask people for formulations that optimize your product. Ask people why. If you don't make that decision or if somebody doesn't make the decision, if you have that decision down in materials management or somebody down in the central sterile areas or something and you're the infection control person, this can help prevent any of those leaking issues and things coming from the OR if they use proper understanding of optimizing their formulations. And then challenge the industry itself. One of the biggest issues I have with a lot of this is that the medical device manufacturer is the person that actually validates your product. The solution manufacturer makes a solution. So they need to talk more. So when you buy something from one person and they have hasn't been validated for this piece of equipment, ask the equipment manufacturer how they know it works for what they've validated, and then ask the solution manufacturer why it's the same as something the other company has validated. So get that data. They have to have data. And another comes is what takes a closer look as to how exactly optimization can work. We're working on things to actually detect dirt and other types of processes that you can't see. We call that the invisible soil perspective, but it's now time, and I believe it's overdue, to take a closer look at how you use chemistry to clean and how that chemistry, when used properly, can actually make your cleaning easier and actually take care of a lot of the problems so we don't have them to show up in the OR. So at that, I'm done.